Hello, everybody. My name is Maya Wilson Brown, and I'm in the Josephs Lab at Michigan State University in the Department of Plant Biology. And today I'm going to tell you about changes in the environmental predictors of flowering onset in Capsella bursipastoris. So much of our inference about species invasions and responses to climate change depend on our ability to predict suitable habitat. And this relies on the assumption that species niches don't change. However, there is considerable evidence that many taxa experience niche shifts throughout space and time. Sometimes these niche shifts occur along with range expansions. One such species that has experienced recent and rapid range expansion is Capsella bursipastoris, or CBP, as I will refer to it today. CBP is a weed in the Brassicaceae family that's thought to have originated in the Mediterranean and has since achieved a near global distribution. To be such a prolific weed, it's had to maintain reproductive success in many environments. It is self-fertilized, but it's not a great competitor against other plants, meaning that the timing of reproductive events, like the onset of flowering, is critical. So in this talk, we'll focus on the environmental niche in which CBP flowers, comparing its native European range to an introduced range in North America. So here I'm showing a classification of climates throughout the world. And while climates in Europe and North America are similar, North America extends much further south and a bit further north than Europe does. North America also tends to have slightly hotter summers and more arid environments shown in tan and golden colors, as well as equatorial environments shown in red and pink. So because of the new available climates in North America, CBP has the potential to change its flowering niche to start at the optimal time in these varied environments. So we wanted to explore how the niche for flowering time onset changed between CBP's native range and the introduced range, and if so, what differences in climatic cues can we detect in the shift? So in this project, we used the date of observations of CBP from the Global Biodiversity Information Facility as a proxy for flowering time onset. And CBP displays a latitudinal decline in the onset of flowering in both its native range in Europe and the introduced range in North America. And while photoperiod is consistent across latitudes, as I showed on the previous slide, climate is not consistent across latitudes. So using that same set of observations on the left, I'm showing counts throughout the year from January to December for observations of CBP. And it appears that CBP tends to flower later in Europe than it does in North America. And this could be evidence for a shift in the flowering niche. So if the climate niche for flowering onset stayed the same in the transition from Europe to North America, we would expect that the models during peak flowering onset in Europe, in this case summer or June, would have the highest predictive ability for observations made during peak flowering onset in North America, in this case spring or April. In other words, a European summer is essentially the same as a North American spring. So to test this hypothesis, we generated 12 species distribution models, one for each month of the year, in a Maxent framework that would describe the environment in which Capsella bursipastoris begins to flower in its native range. We took each of the models and projected them onto North America, and finally, we evaluated each model's predictive ability on occurrences of CBP in North America throughout the year. So here I'm showing the predictive ability of all European models on the x-axis from January through December during peak flowering in North America, represented by the orange and blue lines. The horizontal dashed line at 1 represents random predictability, where an AUC ratio of 2 would be perfect prediction. And what we see is that European summer models do not perform well on observations made in peak flowering time in North America. So here I'm showing the same plot, but for the full year of flowering onset in North America. And generally, CBP that's recorded in cooler months, represented by cooler colors, tends to be well described by European spring and fall winter niches. This suggests that CBP is flowering at the same time between its native and its introduced range, but not necessarily in the same environment. So given the evidence that the flowering onset niche has changed between CBP's native range and its introduced range, we were curious to see how they were different. So here I'm showing the contributions to each model from the environmental variables used. On the left, I'm showing models from Europe, and on the right, I'm showing models from North America. And throughout the year in Europe, minimum temperature contributes most to the models of flowering onset, except for November, although there is an uptick in the contributions from precipitation and maximum temperature in the summertime, as well as day length in the fall. And this could be because Europe can have harsh winters, so minimum temperature is a limiting factor, especially in the spring when CBP that has overwintered as a rosette is getting ready to flower. By summer in Europe, maximum temperature and precipitation become more important, potentially because of drought in the Mediterranean climates. In North America, minimum temperature and precipitation contribute most to flowering models in the winter and early spring, and by the summertime, maximum temperature has the highest contribution to flowering models. And these results could be a reflection of North American summers being a bit hotter or the continent generally having more deserts. So there is evidence for a flowering onset niche shift between CBP's native range and the introduced range. And while CBP displays a latitudinal decline in flowering time, it is likely also influenced by water availability. And finally, I will be joining MSC's plant biology department as a PhD student this summer. So keep an eye out for future experimental work to verify these results, as well as investigating the genetic basis of the differences between these populations. With that, thank you so much for watching and feel free to reach out with any questions or comments.